No, it's okay. Thank you. It is my great pleasure to introduce Professor Michael Warren Young. Michael Young was born in Miami, Florida, March 28, 1949. Later, the family moved to Texas, and he graduated from Lawrencedale Bell High School in Dallas in 1971. Then he started undergraduate studies and got a degree in biology from University of Texas at Austin. And then he continued at that university and got a PhD 4 years later, which was focused on research in Drosophila. After his PhD, he moved to Stanford University in California for postdoctoral training. And during this period, he learned technique for molecular biology and became further interested in the subject of circadian rhythms. In 1984, he became an assistant professor in New York at Rockefeller University, and in 1988, I assume the years are right now, he became associate professor, and from 1988, you were named professor. Sorry, I made this up.
I think it was, 78 was assistant professor,
all right.
And 84 was associate, and 88 was professor.
Okay.
I moved along a little faster.
But then you have been appointed vice president for academic affairs, and currently you are provost for the medical faculty. And over the years, you have done research in this area, and in 1984, independent of the groups of Jeffrey Hall and Michael Rosbash, you described the period gene. Later on in 1994, you described another important gene and protein, timeless, and later on also the protein double time. And this has led to further discoveries in the field, and also you have been able to demonstrate that this can be a mutation in these genes and proteins can be coupled to certain sleeping disorders. So, over the years, Michael Young has received numerous prestigious scientific awards. However, I understand that once you have been awarded the Nobel Prize, the list of further potential prizes is dramatically reduced. Sorry for that. So, now we very much look forward to your lecture, Genetic Control of Sleep and Circadian Rates. Please, Professor Young.
Thank you. I think my job today is to keep Michael awake, but I don't. So let me just start. Some of you may have seen this before, but I want to talk about two issues. One, a little bit more about what these clocks are. I like the term that was used earlier. We often hear of circadian rhythm, but circadian regulation, I think, is a term that encompasses a larger area that I think we've come to understand. And I'd like to talk about our progress in understanding a human sleep disorder that is very closely related to the kinds of things we've been studying for so many years in flies. We have a place in New Mexico where my wife and I often go on long hikes in the different times of year. This is a summertime hike. It's an arid environment. It's quite dry. It rains a couple of days a year, but when it does, it's a foot or so that comes down all at once. But this is a small plant that we passed on a hike a few years ago, and I decided to snap a photo because I recognize the plant. This plant has the name 4 o'clock. Colorado 4 o'clock is its common name. And first, I want to just point out this branch so that you can see in the next photograph that we're dealing with the same plant. And you just notice that all these blooms are closed at 2 in the afternoon when we went out on the hike. We come back 4 hours later and we see a very different picture. All the blooms are open. Here's the branch. Again, it's the same plant. there is a circadian rhythm of flower movements in this plant. This plant is pollinated by a moth called the hawk moth. It's a night active, a nocturnal moth. And I believe this is a strategy to conserve water. We're in an arid environment. It takes water to pump out those petals. You also lose, you're opening yourself to loss of water through those petals when they're open. And if you can close them, you can pump that water back into the plant and into the stems and the leaves of the plant and conserve it. So if your pollinator isn't around, what's the point in having the flower open? So I believe this is an adaptation of that sort. Rhythms are all around us, and I'm just going to give you a couple of examples of what we often see. This is work by Karl Von Frisch. He won the Nobel Prize in physiology or medicine in 1973, along with two others working on different aspects of behavior. He studied, his whole life, he studied the behavior of bees, honeybees, and in particular, the dance language of bees. Bees are not only smart, but they have tremendous ability to communicate with one another. And it's a social system. You're so closely related. Members of the hive, most members of the hive are so closely related that they're almost like cells in your body. If you don't work together, you're not going to make it. And what Von Frisch recognized were two kinds of dances that were involved in communication about the position, the location, and the distance, well, the location of food. One is called a round dance, which is a simple dance that goes something like this. And that dance, this occurs in a dark hive. The dance floor is the honeycomb, which is vertical in the dark hive. And it's a sense of touch and chemical cues that allow other bees, other potential foragers, to follow the initial forager to get information about where the food is. So this would be, the lead bee would be the bee that knows where the food is. This dance just means food is close by, go outside and find it. Not much information other than you don't have to work very hard to find the food. A more complicated dance is this figure 8 dance, which is called the Waggle Dance. And this is because every time the forager that knows where the food is crosses this center point in this figure 8, it vibrates its wings and its abdomen. And the amount of time that it spends doing that and transversing this part of the figure 8 is an indication of how far to the food the other bees must go to find what that bee had found. The other piece of information in this dance is the orientation of this dance on that vertical hive indicates the direction relative to the position of the sun where the other bees should fly in order to find that food source. So for example, if the food is in direct line with the position of the sun, it's a vertical dance like this. If there's a 40-degree angle, let's say, to the sun, then it will be offset by 40 degrees in a particular direction. The problem, of course, is that these dances can go on for minutes and hours, and that foraging bee doesn't go out again to find the food. And yet, in this dark environment, it has to give information about where the food is relative to the sun. And of course, that's an inconstant. The apparent location of the sun is changing. And what Von Frisch found was that over time, the original forager changes the angle of that dance to correctly anticipate the position of the sun so that it's giving useful information to the rest of the hive about where to forage. We know now from work by Steve Reppert that a similar system takes place in migrating monarch butterflies, and the clocks that are involved, they're circadian clocks of the sort that Michael was telling you about, are located in the antennae. So they're not in the brain, they're in the antennae. You can just paint the antennae and you can destroy the ability of these animals to find their way. Much more familiar is a rhythm of this sort. This is a sleep-wake or an activity rhythm from a hamster. This is from work done in 1960. This was published. This is 25 days in the life of a hamster. Each one of these lines is a 24-hour interval. And in the same way that Michael just told you about generating deflections when the animal's active, these are deflections caused by movement of a running wheel. Hamsters will run miles every night. They love their running wheels. And you see evidence for that here, where they're in constant darkness for 25 days, and this one animal is getting on and off that running wheel with tremendous precision. A couple of minutes a day, you can predict with that kind of accuracy when the animal's going to get on and off of that running wheel. And another feature is it's not vertical, so these aren't 24-hour rhythms. They're circadian, or about a day. And so in this case, they're slightly shorter. The internal rhythms are slightly shorter than 24 hours, and it's hard to imagine that something like this could be anything other than endogenous pacemaker of some sort. In the 1970s, there were efforts to localize cells responsible, parts of the brain responsible for this kind of behavior. Lesioning studies followed these kinds of observations, which were that if you provided a radioactive form of glucose to the animals, there would be different metabolic activities that would show up in the brain, and specifically high levels of difference day and night in a region called the SCN, which is a part of the hypothalamus. Later, surgical experiments would ablate this region of the animal's brain. The animal would show activity in sleep, but arrhythmically, so that they lost this coherent rhythmicity in the absence of that portion of the brain. Many years later, as Michael pointed out, Seymour Benzer and Ron Konopka at Caltech asked a similar sort of question, which is, if cells can control, or if a particular part of the brain can control this kind of rhythmicity, cells are there and functioning because of genes they contain. Are there genes? Can we take this down to genes and ask, are there specific genetic elements that are responsible for these rhythms? And as you see here, and as Michael explained, they found 3 different mutations that had profound effects on the rhythm of the fly: an arrhythmic mutation, a long period mutation, a short period mutation, fast and slow clocks, as well as the loss of clocks from this genetic screen. These turned out to be point mutations, which I became very interested in as a graduate student, really by accident. I was working on a question about gene density in a small region of the X chromosome of Drosophila. It happened that their preliminary mapping suggested that these mutations would fall close to the region where I was working as a graduate student. So an interest in behavior and the novelty of these mutations got me to ring Ron and Seymour up, ask them for the mutations, which they sent immediately. As I said, I'd like to see if I can use some of our materials, our tools, to see if I can map them more precisely. So this is a part of the chromosome we were working on, and these all represent breakpoints of chromosome rearrangements. And at the time, what we found was that rearrangements with endpoints in this area here all circumscribed Ron and Seymour's mutations. So we knew that the gene was mapping somewhere here. One of the things we had at the time were lethal mutations throughout this interval, a number of different vital complementation groups. It was clear that the permutations were not a part of any of those previously known genes. So this wasn't, that was an important realization, that these weren't just viable forms of a deeper physiological, or another kind of vital physiological problem. And in fact, we now know that the loss of function is arrhythmicity and not any of these other phenotypic features. But most important was this translocation, which I had originally assumed had no phenotype whatsoever, but it failed to complement Ron and Seymour's mutations and altered the region simply by joining one part of the X chromosome to the 4th chromosome and reciprocally. So that was of tremendous interest to us, because later, as I went on and learned molecular methods for cloning DNA, it was clear that if you were going to walk through thousands of base pairs of DNA, you're not going to be able to find a point mutation in 1983, 1984, but you can't miss a chromosome rearrangement. And in fact, we and Michael and Jeff did just this in 1984. This is a number of years later, of course, and we found that there was a single region within this long chromosomal walk which could be moved into a fly that had no rhythms. And if you removed a single gene from a wild-type fly, micro-injected it into flies that had no rhythmicity, you could restore rhythmicity. So that's essentially where Michael and Jeff and I began. What is this thing that when you give it to an arrhythmic fly will bring rhythms back? And Michael's talked about some of these things. One of the surprises, we were disgusted initially to see that this was active in multiple tissues. We thought we were working exclusively with behavior. We now know that there's much more significance to these clocks, but at the time we thought something's wrong. This is supposed to be behavior. We later learned from DNA sequencing that the 3 mutations that were found by Konopka and Binzer were affecting the structure of the protein. Michael and Jeff found that the RNA and protein cycle with a daily rhythm, and also found that the mutations feed back to alter those protein cycles. And as Michael pointed out, there was a great deal of evidence for some kind of a feedback loop in which the product of PER was feeding back to inhibit its own production. One of the things that we wanted to try to understand was, why is this an oscillator rather than a thermostat? So if I go over to the thermostat on the wall and set it to 70 degrees, we don't swing between, and I'm talking about Fahrenheit now, so that nobody has to, sorry. But if we stick with Fahrenheit, we don't have a system that will swing between 100 degrees and 30 degrees. We have something that feeds back in a very deliberate fashion to be a homeostatic mechanism. Feedback in that case is meant to hold things at a constant. In this case, we've got wide oscillations. We want to build an oscillator. So how might that occur? And to understand this, we needed to find additional components of this machinery to understand how that oscillation might occur. And it was tough going at first. This is Photoshop. This is not real, but it does reflect. Konopka and Binzer screened 200 flies to get the first PER mutant. And we thought we might have a chance to be lucky pretty early, but it took 7,000 bottles of flies using the same method that Konopka and Binzer looked at. They looked at the eclosion pattern, the hatching of adults from their pupal cases. You have to do this multiple times a day with each bottle and plot the numbers of flies that are hatching. They usually hatch in the morning. We finally found a bottle of flies in which there was a regular hatching, and that turned out to be a mutation of the gene that we called timeless. Timeless, as you'll see, and as you heard, wound up being a partner for PER, so we immediately were struck that the very second gene in the system took us back to the original genetic problem, which indicated to us for the first time that there might be just one clock system in this organism to be understood. Thanks to Michael, who invested quite a lot of money to induce the production of machinery that would allow large numbers of individual flies to be monitored, their locomotor behavior, and he's already described how this device works. But we were able, we and Jeff and Michael were able to screen thousands of flies per year in order to really provide a genetic saturation for this system. And as a result, 10 or 12 genes were discovered, and all of their products and activities made sense when we put them all together, when we compared notes between Brandeis and Rockefeller. So these are examples of the records for mutants. These are all mutants of a gene that we found called double time that Michael mentioned. Heterozygotes give an aberration, homozygotes even a more extreme aberration. And of course, each gene, we had multiple mutations, multiple alleles, and we get both short and long period variations, very much like we had at the PER locus coming from Ron and Seymour years before. So the way all of these aren't all the parts, but these are key players that I think can be used to describe the way this machinery works. The two genes, period and timeless, are on different chromosomes. They're activated by clock and cycle, so transcription is promoted by the association of these proteins with PER and with timeless, so that we have synchronous production of period and timeless RNAs that should give rise to period and timeless proteins. At the beginning of each one of these cycles, however, a partnership occurs very rapidly with newly synthesized period proteins, double time, ortholog of casein kinase 1 phosphorylates PER and causes its rapid ubiquitination and destruction. So double time is constantly produced and is at high levels. That's the first dance partner that PER sees at the beginning of each cycle. So we make very little period protein early in the cycle because of the action of double time. Later, as timeless proteins accumulate, we can now have a different partnership with PER, and that is a PER-TIM partnership, as Michael mentioned. And that partnership does two things. First, the timeless on its own and period on its own seek the cytoplasm. They spend most of their time in the cytoplasm, but as a complex, they move to the nucleus. And in the nucleus, they physically associate with clock and cycle and abrogate their activity as transcriptional activators. So the net effect of this is that double time is producing a delay in the accumulation of period, and by delaying the timing of this inhibitory process from the formation of RNA by these two genes, we introduce a delay that will produce oscillations in the system rather than a homeostatic regulation by this feedback. Michael had mentioned that this is a system that responds to the environment. Light is probably the first and foremost environmental signal that life on Earth had to contend with. And in this case, light is received by a photolyase-like protein called cryptochrome. And when it is excited by light, it physically associates with timeless. Both proteins are phosphorylated and degraded. So all day long as the sun is up, as long as light is shown on the system, timeless proteins, even in the presence of vast amounts of timeless RNA, will not accumulate because of this light-dependent destruction. And of course, if there's no timeless accumulating, double time is going to be the only partner for period, so no period will accumulate. So all day long, you have no period or timeless proteins. When the sun sets, you have all of this RNA that can now be translated into protein that can close this cycle. So it's easy to see, I hope, how a light-dark cycle can feed into the system to set the correct phase of the clock. So when I go from New York to Stockholm, or when a fly goes from New York to Stockholm, it would be reset by this kind of a mechanism. As Michael mentioned, these are largely conserved mechanisms. So there are 3 different period proteins in mammals, shown over here, instead of the 1 in the fly. Clock and BMAL are orthologs of clock and cycle, essentially the same proteins. Period is regulated by CK1, as it is in the fly. And one variation, an interesting variation, is that cryptochrome, which here is a regulator of timeless accumulation, is now the partner for PER. So it's part of the inhibitory complex that goes to the nucleus and associates with clock and cycle, and that'll be important later in the discussion. But you can see this is over hundreds of millions of years, tremendous conservation across the animal kingdom. These clocks, as Michael pointed out, control a large number of genes and in many different locations. This is an assessment of genetic gene activities in the Drosophila head that were done. This was done about 16 years ago now. And what this is every row is a gene with red and green indicating highs and lows of expression. And those at the top, those genes at the top are starting up, have their peaks at around dawn. Those in this area around noon, peaks at dusk are in here, midnight in here, and so forth. And this is over a 6-day interval where sometimes there's a light-dark cycle, but half the time there's not. And what you see are 6 stripes of red and green, which indicates there are waves of gene expression occurring. The other thing is that these are diagonal stripes, so I think, again, as Michael pointed out, these are this is not a clock that just controls nighttime versus daytime genetic activity. You've got unique relationships of these oscillations at each hour of the day. So the fly can know what time of day it is. If it can assess the combination of gene activities, it does know the time of day, essentially, through that combination of gene activities. This is the same plot again, but I want to point over here to a different experiment in which we're now looking for those oscillations in 3 mutants. This is timeless, this is period, this is clock. Those are arrhythmic mutants, and what you see is all the gene activity. The gene activity is no longer rhythmic. It's cacophonic, if you will. And this is I think something that strikes me about this as important is that these are adaptations to a cycling environment. We'd never have these clocks if it hadn't been useful hundreds of millions of years ago to pay attention to these environmental signals. But now, with these clocks highly evolved after 2-3 billion years from the very first ones, the environmental signals can no longer have an impact on gene expression unless there's a clock in place. Everything is feeding through the clock to organize temporally these gene expression patterns. So that's maybe surprising, but I think a fundamental lesson about the position that these clocks take up in our biology now. So just a quick summary. I'll let you, just for interest of time, just read through these yourself. But those are the points that I've made. One of the other points to be made, in 1997, Jeff Hall and Steve Kay and their colleagues did a study of Drosophila in which they used a luciferase reporter to look at what appeared to be the consequences of period expression in different parts of the body. And as Michael pointed out, in the earliest of days, we looked at where PER was expressed in the fly and were quite confused by the fact that it was abundantly produced in many, many locations. But it wasn't clear at that time that those were active clocks. But what Jeff and Steve found was using a luciferase reporter that could be monitored in real time, either in whole flies or dissected tissues, where that there were clocks all over the animal that were oscillating and would oscillate in a dish even when in a dissected fly. So this indicated, flies are translucent. All these tissues had their own photoreceptive mechanisms that I've described previously. So in a fly, you've got these fiercely independent clocks all over the animal that all want to tell time by themselves, and really, on occasion, will listen to each other, but not necessarily. And in mammals, we have a similar sort of thing, where there are clocks all over the body. Our skin has clocks. We've used that, and I'll talk about some work where we've used that to monitor circadian rhythms in humans. But our skin can be put in culture. We can put reporters in, and we can see these very nice, robust oscillations. Our liver, our lungs, our skeletal muscles. There are a few tissues where clocks have not yet been found, but in most cases, we find clocks just about wherever we look in a mammal. And so I want to use that as a way of asking questions about what happens when we have, how do those clocks get their information? How do they know what time of day it is? How do they know how to coordinate their activities with other parts of the body? And what happens if we introduce a conflict into the information coming into the animal? And so a number of years ago, a colleague, Michael Minneker, who was at the University of Virginia, did an experiment in which he tried to confuse the animal. Mice or these were rats, are nocturnal. What would happen if you take an animal and give it food only at times when it should be in the middle of its sleep interval? And so what Michael did was to take a fly on Eastern U.S. Time zone, where all the clocks should agree. It was if it's 1 in the afternoon, then it's 1 in the afternoon by the readouts of all of these tissues. What would happen if we for several days feed this animal in the middle of the time when it should be sleeping, which is the middle of the day? And after a few days, the animal learns to do this. It produces a modified pattern of activity where it'll get up for an hour or so, eat the food, go back to sleep, and then get on its running wheel all night long. So already at the behavioral level, it had a modified pattern that was pretty interesting. What was learned was that after several days of that kind of a pattern, the brain is still on is still in the same time zone as it started, but these other tissues moved in many cases dramatically away from the phases that they were assuming when they were coherently together in an animal that was feeding and active simultaneously. So we can interrupt these clocks. What this appears to be telling us is that many of these clocks are not watching the light-dark cycle. They're watching when fuel is coming into the system or watching some other organ that is reset by fuel coming into the system. And we produce a desynchronization of this sort. Now, these animals are not very healthy after a few weeks. And in fact, some of the difficulties that you see here resemble what happens when you start specifically in a tissue-specific fashion knocking out the clocks in these individual locations. And some of the kinds of difficulties that wind up when you knock out the clock in specific organs are pretty striking. You knock it out in the pancreas, you get a mouse that starts looking down. You knock the clock out in the liver, you can produce metabolic syndrome. So the consequences of not having coherence is apparently very important and something that deserves a lot more study. So I now want to move to a study of humans, and I want to ask a related question, which is, this is a deliberately desynchronized animal. And in fact, something like this is probably similar to what Michael and I have gone through coming from New York City. We're at least the first few days after arriving, we've been here long enough that I think we're settling in, but the first few days after arriving, we're really occupying multiple time zones simultaneously because our clocks don't move coherently to a new time zone. They have different rates of reaccommodation, and so we're very much like this mouse for a few days. What happens if our internal timer doesn't agree with the outside world? I mean, that's what we have with a per S and a per L mutant in the fly. That's a conflict with the 24-hour external world. And so we've been studying something called delayed sleep phase disorder, DSPD for short. And you can think of individuals that are affected by this disorder as night owls. So given this artistry that the university, Rockefeller, drew up a few months ago, you can imagine someone tapping away late at night. I'm sure this is a familiar picture, right? Up late at night at a maybe even worse, a computer screen. So you're getting reset information. In addition to being up and active, getting reset information from light coming off these blue emitting computer screens. So delayed sleep disorder is pretty common. Best guess is it's probably about 5% of the population is affected by this. And it's a chronic, we're interested in a chronic problem. That is something that subjects tell us has been with them for as long as they can remember. A persistent delay in the major sleep episode and resistance to efforts to advance sleep phase. And here's an example of a DSPD individual on the right and as compared to a control individual on the left. This is double plotted, so we've got 6 days of recordings over here, and the same 5 of those 6 days are also shown over here. The reason for that is we want to look at the interval where the subject is sleeping, and we're doing 2 things. They've got an activity watch so we can see when there are motor movements, and we've asked them to put a mark in a logbook when they get out of bed and when they get in bed. And those 2 things are agreeing. The in-bed time is a red triangle, the out-of-bed time is a blue triangle. And you can see this individual is going to sleep every night around 11:30, getting up at, 7:30, maybe 8 o'clock in the morning most days, and has a very regular pattern. On the other hand, we've got this ragged pattern coming from this DSPD subject, Tau 11, that you'll hear more about. And the in-bed times are all over the place, all well after midnight, some just about dawn. And the out-of-bed times are coming pretty close to noon. So this is a severe case of a phase difference that we'd like to understand better. This is a part of the kindred that this Tau 11 subject is a part of. This is Tau 11. And these are relatives, a cousin over here. And several of the relatives colored in either black or gray had what appear to be aberrant patterns of sleep. I'll explain these numbers in a minute. This cousin over here has an apparently normal, more common pattern of sleep. We did whole exome sequencing on all of these members of the family to see if there was something that was following the segregation of this disorder. We found something that appeared to be behaving like an autosomal dominant from the whole exome sequencing, and that was a mutation in now a gene that's familiar to you, cryptochrome 1, which is the partner for PER in that transcriptional inhibitor. It's a point mutation. It's a single change, but it's at a splice donor site in the gene, and therefore we skip exon 11. Exon 12 is spliced directly to exon 10, so we miss 24 amino acids in the protein. That's shown here. This is a series of subjects that were in this study, and here's Tau 11. She's heterozygous. She makes 2 forms of that protein. We know that a gene is capable of changing rhythms because we can drop it into a cultured cell that has no cryptochrome, restore rhythms, but restore rhythms that differ depending on whether you give back a wild-type copy or this variant form of the protein. So here's the wild-type copy of the protein. This is the variant form, about a half an hour change in the cellular rhythms when we give that version of the gene back. So clearly, this one change is capable of altering rhythms at the cellular level. Just a reminder again of where cryptochrome fits in. It's the partner for PER, part of this inhibitory complex. So its job is to bind to clock and cycle and turn them off. And here we're looking at the affinities of the 2 versions of cryptochrome, the more common version of cryptochrome versus this variant form. And if we start down here, where we're doing immunoprecipitations, for example, here are input proteins, either full-length protein, cryptochrome, the truncated form of the cryptochrome protein, or a synthetic heterozygote, equal amounts of these proteins. you see that if we immunoprecipitate clock, which is a target, the full-length protein is a weak interactor. We pull down much more of the truncated protein, and in the synthetic heterozygote, primarily that truncated form of the protein. Now, we don't see that kind of a difference when we ask, how does this version of cryptochrome interact with PER? So apparently we can make the inhibitory complex just as efficiently as with the two wild-type forms of the protein, but when it comes to that complex interacting with the activators, we have a big difference in the affinity, which we believe may be closely connected to the fact that it's a dominant mutation. We were surprised when we looked at databases at the frequency of this variation that it's quite common. So the overall, especially considering it's a dominant, then it's the carrier frequency that's important. So twice this would be the carrier frequency, indicating that if we just lump every population together, it's about 1% of the population. In Europeans, about 1 in 75 descendants of European ancestry are carriers of this mutation. And that allowed us to say we should be able to find other subjects to have this, that have this. Can we find them and can we characterize them? Does this mutation predict the presence of DSPD? And in fact, these are just examples of a few families. Again, carriers are dark, and open circles and squares are non-carriers. And here on a clock face, we've plotted the mid-sleep points, the time of mid-sleep, 4:30 in the morning for most of these, for the non-carriers, and then the similar mid-sleep points for the carriers. And what you see is most of the carriers have delayed sleep by 2.5 hours relative to the controls. But we also have another phenotype, which are nappers. They have fragmented sleep that shows up as a series of naps. So we have interesting two phenotypes to study in the future. So I'll just summarize very quickly. We've got several unrelated families that were carrying this mutation, predicts DSPD. Since it's found in multiple members of each of these families, then it's running through the families in a hereditary fashion, so all the organs must be affected by this. We're very interested in what other consequences might be. Enhanced binding to the activators suggests basis for dominance, and we have seen that we can reprogram circadian rhythmicity in a single cell by transferring this gene to a naive cell. So I'll just end by looking ahead. What are we thinking about now? One of the things that are of most interest to us is that sleep disorders are commonly associated with a variety of metabolic and other psychiatric issues. So are there causal relationships between these? I don't think we've really had a way to study this in the past, but if you have large families, all of which are genetically identical with respect to a single variation, you should be able to sort out environment from genetics and really answer this question about whether certain disorders are connected in that way. So I'll just finish by pointing out Alina Patke, who's done almost all of the work that I spoke about with regard to human rhythms. She initiated each of those projects, but with the other individuals named here, was able to, through collaborations, get all of that work accomplished. So just a thank you to Alina. And thank you to you for inviting us and being such enthusiastic hosts. Thank you very much.
Well, there is no time for questions, but I have to ask one question. You say you didn't find a clock in some tissues yet.
I think the spleen, for example, has been claimed. People have looked in the spleen and have not found evidence for clock activity. But we can be fooled. For example, in the intestines, intestinal epithelial cells will show a rhythm, but only if but will not show a rhythm in a germ-free animal. So there are interactions that we don't yet, there are facultative rhythms that show up in many cases. So I think this is another interesting area for exploration.
Okay, thank you very much to both our Nobel laureates, and we look forward to have you coming back even more to Uppsala and Scandinavia, and hopefully we can learn more about this very intriguing field. Thank you very much again.
Thanks. Thank you. Thank you.
Now we have to proceed to the
Sure.